I love parafilm. But today's post isn't about parafilm. Today's post is about agarose gel electrophoresis, which is this technique that we can use to separate pieces of DNA by size. And so there's a trick I could use with parafilm to prepare my samples, and I really just love any excuse to play with parafilm, which is kind of like this super thick saran wrap type stuff that we can use for like sealing things or doing all sorts of things. In this case, helping prepare our samples because it's all waxy, and so you can like mix things on it with little droplets. But anyway, so agarose gel electrophoresis. So like I said, it's this technique that we can use to separate pieces of DNA by their size, so we can look at them and see what's in there and that sort of thing. And sometimes also so we can then purify them out of the gel. So electrophoresis, use electricity to get things to move. And in this case, we're moving them through a gel. And this gel is like this mesh. So you can think of like the DNA pieces as like jump ropes. And then this mesh is kind of like a sea of basketball rims. And you're dropping the jump rope through the sea of basketball rims, and they're gonna get all tangled up in the basketball rims, right? But they're still gonna want to go through because, well, in that example, it'd be like gravity, but here you've got that electricity really pulling things along, and because you have the negative charge attraction that we'll talk about, because DNA is negatively charged already because it has this backbone with all these phosphate groups, and so that negative charge, when you set up this like electricity thing, so you're gonna set this up. So you hook it up so you run to red, so you're running to the positive charged end. And so your DNA is negatively charged, you stick it in the little wells at the end of the gel, turn on the electricity, then they'll get attracted. It's a whole opposite charge thing. Charged things attract, like charges repel thing that I've been talking about so you get that repulsion from the top and the attraction from the bottom, and it goes through. Electrophoresis. And then that gel is going to slow them down differently, and then when you stop them, like the bigger things won't have traveled as far because those longer jump ropes are more like tangled up in those basketball rim hoops. And so then what we can do, so you have the bigger things up top and the smaller things on bottom, and then we can use like a fluorescent stain. So basically fluorescence is when you have a thing and then you can shine light of one length at it and it'll give off light of a different wavelength. And so we use a stain that can bind to the DNA, and then we stick it on like a fluorescent UV screen tray thingamabob, and it'll shine light, and then it'll give off light, and it'll take a picture for us. And so then it'll look something like this, so here's like a ladder. So it's like you buy this pre-made. It's like a mix of DNA of different sizes, and then you can compare the sizes of your thing. So here I was doing some cloning, and so basically I was trying to get a recipe for a protein I wanted into a vector, so like a circular piece of DNA. So in this case, it was like I was using a SLIC method, which is this PCR-based method. So basically, PCR, you make lots and lots of copies of a DNA, like of a specific region of DNA. So you specify the beginning and the end, and then it'll make copies of the part in between. But then you want to see if it actually worked, and so you can use agarose gel electrophoresis to separate the pieces and look and see. So you have, like, I have like the vector pieces, so the like both part of the thing I want to stick the thing into. And then the thing I want to stick into that thing. So it's smaller, and so you can see it's down here. So electrophoresis, that word might sound similar to you, or at least it will if I tell you it's the E in SDS-PAGE, which is a technique I talk about all the time because it's all the rage, right? So SDS-PAGE is the technique that we can use to separate proteins by size. And so it's done in like a contraption like this, but with both of them we can set them up and hook them up to like the same type of power box, but run them at slightly different voltages and that sort of thing. But that's not the biggest difference between them. There's a lot of differences between them. But so with SDS-PAGE, we're separating proteins. With agarose gel, we're separating DNA. With SDS-PAGE, we have these thin gels that are like in between these plates, and it's running vertically, so you're going. With agarose gel, it's like this horizontal slab, and it's going or whatever towards the red. You just want to make sure you hook it up right, or else your sample's going to go out because, yeah, the charge thing. But anyway, so someone asked me once, like, why is SDS-PAGE run vertically and agarose gel run horizontally? And I think it's because, like, with SDS-PAGE, you have this really thin slab. And so you have to put it between these glass plates. And so then it makes sense to just, like, do it like that. But with Agarose gel, you just have this thick slab, and so it's like stable, like if you just lay it down, so you don't need to stick it between like glass plates or anything. And so if you were to I mean, it's like pretty stable, but like if you were to try to stand it up without like plates or anything, it would just, so you want to do, you just like stick it down when it's horizontal like this. So right now this is actually in the casting position. Like this. So this is when you when you're preparing it. So basically it's the gel. We're gonna go way into more of all this in detail. But the gel's made up of the sugar called agarose. And so we just keep this big thing of it here. Agarose, we typically make like 1% gel, so we put like a gram of this into 100 mils of TAE. So we've got this whole setup because we run a lot of these. And then you actually, so you have this sugar powder and then it dissolves. So you heat it in the microwave. I typically do it in like 15-second spurts or so, just so you don't like overboil it. But yeah, so, and then you do that in the microwave and then it'll be like this hot liquid. And then you pour it in here. If you don't have a casting one that goes like this, you can like take the edges of the gel of the casting block or whatever, but make sure whichever way you do that it's really tight. So if you're new to it, you might want to try doing it with water first, just to see if it leaks before you try putting your agarose solution in there. So, and then you put like this comb in it, and then it's actually gonna gelidify. So with the SDS-PAGE gel, it's like polyacrylamide, and you have these like long polyacrylamide chains that you actually like get to polymerize. So the chains actually like covalently link, so they in really strong bonds, like the type of bonds that make up molecules. Whereas with agarose gel electrophoresis, it's more like knitting yarn, and so I'll talk more about that sort of thing later. But basically it will solidify on its own, like you don't have to add anything. When it cools down, it's gonna like gelify. Quick word of caution, so like when I was in undergrad, there are different types of agarose basically, and there's like low melting point agarose, which is what you use like if you want to actually like purify the thing out of the gel afterwards, which can be really helpful if you're trying to like separate pieces from like a restriction enzyme digest or something, where you have different size pieces and then you want to take out one of those pieces. So, but the thing is, like, it's a low melting point, so the idea is that it's easy to get things like out of it afterwards, but it's also like floppier, and it's like way more fragile. And when I was in undergrad, I didn't realize I was using the low melting point when I was making like all my first gels, and I just thought it was like totally normal that gels were that crap, like, and I was like totally just like hated them because they was always breaking, and then I was like, oh my god, I was using the wrong agarose. So yeah, read your labels and be careful about that. But anyway, another thing to be careful about is, so we have this like mitt thing to help us with the don't burn ourselves. And undergrad or at least I couldn't find one, so I'd actually like fold up a paper towel and use that to kind of like pick it up without actually touching it. But whatever you do, be careful. Probably best practice wear goggles too. Anywho, okay, so that's one difference between SDS-PAGE and an agarose gel, or I guess that was more than one difference. That was several differences, but another key difference is that with agarose gel electrophoresis, what you're doing is, so you have with either of these techniques, so SDS-PAGE or agarose gel, you're gonna have you need the molecules that you're gonna separate to be negatively charged, and like you want them to be consistently negatively charged. So with proteins get their charge from the like unique parts of protein letters. So protein letters are called amino acids. They have this unique, each of them has like a generic part like that hook together to form this chain, like this backbone, and then they have these unique parts called side chains or R groups that stick off. And some of those groups are like negatively charged, some are positively charged, most are neutral. But different proteins have different combinations of these letters, and so they're going to have different charges. And so some proteins might be negatively charged, but some are positively charged, some are neutral. So, and it's not consistent. So like a big protein might have like really high charge or whatever, and low is, you know, it's like, so it's all over the place. So with the idea with the SDS-PAGE is that, so the SDS part stands for sodium DodecylSulfate, which is this detergent, so like an artificial soap. And what it does is it's like, has a bunch of different roles, but it's going to, so basically you heat up your protein to help like denature it, to unfold it, and then this SDS will come in and like slither in, and it's going to coat the protein so it doesn't like refold, because now it's all like slithery. And this like SDS is negatively charged, and it coats the protein uniformly, so you have this like long strand of protein, and so now you have this charge to length relationship. With DNA, it's built in. It's got these phosphate groups in the backbone. So it's got this consistent charge, because those base parts, so the unique parts, so like the A, C, T, G, or whatever, that allows them to form those base pairs to keep the strands stuck together, those are neutral, no charge. So, but the backbone is charged, so we don't have to add anything. It's already negatively charged, and you've got this built-in length charge ratio. So that's not gonna like mess up your whole like separation thing. So yeah, with either of these gels, you can kind of like customize how thick, I mean like how thick, but also like the meshiness, so like how close together all those basketball rims are. And so the basketball rims, like, so with Agarose gel, they're typically less consistent and like bigger. So with the SDS-PAGE gel, they're like smaller holes, whatever. So it's like you get a finer resolution of how well you can separate different size things. But with either, like the percentage of the gel, so like how much there is of like, how much of it is kind of just like water, because it's a gel, so it's not just like a solid, there's like liquid flowing through it. And so in this case it's a buffer, so it's like a salt stabilized pH stabilized salt water, and so that's going to help like keep things steady and like conduct the electricity through it. So really it's kind of just all of these base basketball rims are like in a sea, so it's kind of like it's kind of jello-y, the gels. Well, with an SDS-PAGE, yeah, it's really thin too, so it's but with this, it's kind of just like this slab of jello. But you can use like a higher concentration gel to separate like smaller things, because if you have two hoops that are like really far apart, then if you have little things, they're not gonna like they won't be different between the two, because they'll just kind of like both go through a hole, you know. So it's like, well, which one's smaller? But if you have a lot of them, then even the small things are going to separate better. But the bigger things are kind of, they'll just get all like stuck at the top, or they won't separate that well, because then you have like the opposite problem where they're all just getting stuck really a bunch. So yeah, so now let's go look in detail. Okay, just a note that I'm going to be like repeating some things in like different forms, like, so like me talking like today in September form, and then like me talking with slides in June form, and they're not going to be that greatly combined or whatever, because I'm just taking a break from thesis writing, but I wanted to give you guys this resource that you could use, and I miss playing with parafoam. This is a technique in which we can separate DNA fragments by size. It's really helpful for if you've done things like a PCR reaction you want to see works, if you want to see if you do like restriction enzyme digest, so you're cutting pieces of DNA, and then you want to see the sizes of those pieces. So there's a lot of different uses of Agarose gel electrophoresis, which we normally just like say like Agarose, like I need to run an Agarose, like or an Agarose gel. So basically the basic idea, and then we'll get into the details more, is that the DNA has a negatively charged backbone, so you can use that to send it traveling through a gel towards a positively charged end. So this is electrophoresis, so using electricity to get the DNA to move. So the DNA is going to want to move because of that charge, but the gel mesh is going to make the DNA get all tangled up as it tries to travel through. And this means that when we turn off the electricity, so you stop having that force propelling the DNA through the gel, then and you look to see where the fragments are, the bigger pieces won't have traveled as far because they're going to get tangled up more. It's kind of like, I like to think of dropping a jump rope through a sea of basketball hoops. So the smaller fragments are going to be lower down, the bigger fragments bigger up and farther up, and you can run like a size ladder of known DNA pieces of known size to get an idea of the size. So if you've been following along the past few days, this might remind you of polyacrylamide gel electrophoresis. So we looked at SDS-PAGE the other day, which is a similar technique in which we could separate proteins by size. There's also a technique called urea-PAGE where we use a gel to separate urea-coated RNA by size. So I'll show you in a minute a couple of the differences. So the main difference is that I'm showing here is what the gel is actually made of. So with Agarose gel, it's made up of agarose, which is a sugar. And with PAGE, it's made up of these cross-linked polyacrylamide chains. So the cross-link means that these acrylamide chains are covalently bound. So they're really like tightly attached to one another. And so when you get it really hot or anything, it's not going to melt. You can also form a finer mesh than with agarose. But the really cool thing about agarose is it's kind of like yarn that you're knitting, the yarn like self-knits itself, but you're not actually making permanent covalent bonds. And so with agarose, you're not actually like cross-linking anything. You are there's no polymerization steps. It's just like a gelification. So what we do is we actually like make a solution of agarose and we heat it up in the microwave in a little flask to get it to dissolve, and then we could pour this gel and then it'll harden. It's really cool. And so basically it looks kind of like this. And so a key difference other than the type of thing that they're separating is that with an agarose gel electrophoresis, you have this horizontal slab. It's hard to see in this picture, but it's probably like half an inch thick maybe. And with an SDS-PAGE gel, you have a vertical format and you have this really thin gel that's actually, it's held in place by like glass plates or plastic plates. And so you're actually running it vertically. And so with either way, you're going from a positive from a negative charge end to a positive charged end. But with the SDS-PAGE, you do it vertically. And in this case, the gel is being held up between the plates. With the agarose gel, you don't need plates, so you can just like lie it down horizontally onto this tray. And then, so for the visualization, so with the nucleic acids, for like the agarose gel, you normally do a fluorescent stain, as I'll show you. Whereas with proteins, I'm showing you this was a Comassi stain, so a visual colorimetric or like color readout. But the basic idea is the same, that you have these things traveling through the gel and they're going to get slowed down more the bigger they are. But a key thing about the difference between SDS-PAGE and nucleic acids, other than what we've talked about, is that proteins are made up of amino acids, and there's 20 different common ones, and some of them are charged and some of them are not charged. And so your proteins are going to have various charges. whereas with nucleic acids, so for DNA and RNA, you have a negative charge built into this backbone, and all of the so there's 4 letters and they all have the same negative charge. They're unique parts, so the nucleobases, those don't have any charge. So it's the backbone that has this charge, and all of nucleic acids have the same length to charge ratio. This is not the case with proteins, where you have various charges. And so for proteins, you actually have to give them the negative charge, which you can do with the detergent SDS. And the detergent also helps keep it unfolded and soluble. So yeah, so like I said, you have this gel that's made up of this Agarose, and I just thought this was really cool when I learned how it actually works. So I have a post with lots more about that if you want to learn more about the Agarose, but yeah, it's really cool. So we don't need the SDS in our sample buffer, but we still need a couple other things. So by sample buffer, I just mean like the thing that we mix our DNA sample with before we stick it in the well and then turn on the electricity. A key thing is glycerol. So it is basically, it just makes it heavy. So it doesn't just float out of the well. Even still, you want to make sure that you're like pipetting slowly. So you don't get like bubbles that just like pop out when you're trying to pipette it. So slow and steady wins the race. Yeah, so the gels have these wells that you stick your sample into, and then you can run through. Cool thing about Agarose gels too is that you can kind of like reuse them. So if something has run through it already, you can still run something through it. And you can use like partial gels. So you can actually, like, if you only use half of a gel, you can cut it off and then someone else can use it. So yeah, so in our lab, we normally like keep gels going. Like there's just like a common gel out there, and like if partial gel or whatever, and you just make sure no one else is planning to use it right then, and then you could use it, and they last for a pretty long time as long as you have buffer over them. Buffer is just like in this case, buffer is just like a pH stabilized salt water, so often TAE. Another great thing is that you can like set these up to have like 2 combs in them, so then you get like a ton of wells. And so you can actually, what you can do is you can run like 2 rows of samples at once, as long as you stop it before like the first row gets into the second row, which you can tell based on that dye. So the loading, so the dye, the tracking dye, so not the dye that actually stains the DNA, but the dye that's just to say, like, okay, here's where like the liquid's gotten to. So yeah, so that's a cool trick. Just make sure you stop it before you go into the bottom gel. So with that parafilm trick, in the liquid, so you have your sample liquid, which has the pieces of DNA that you're trying to separate, and then it can have different things. So one thing it's gonna have is like glycerol, which kind of just makes your sample heavy, so it'll sink. Another thing it's going to have is a dye. So similarly to how you have a dye, tracking dye in SDS-PAGE, so both of these have these tracking dyes, which is really just gonna let you see like the gel status. Like it's not gonna show you the size or anything. It's not binding to the protein. It's not binding to the DNA. But in the type of sample loading buffer, so like the little sample preparation stuff that we use for the DNA ones, we actually have like a fluorescent dye in there. I think it's called like Easy Vision or something. So basically the fluorescent dye is actually going to go into like and intercalate with the DNA usually. So like this dye is going to get in between like the different, like the rings in your DNA ladder. And then, but you're not going to see that dye, because that dye is fluorescent, and so you won't see it until you stick it on like a UV tray, and then it'll shine light at it, and then the the fluorescence thing will then, that's bound to the DNA, is going to shine light back, and then the camera can take a picture of it, and then you can see it. So that's another difference between SDS-PAGE and the DNA. So, but either way, you're typically doing like an all protein stain. So with something like Comasi, but that's like a post stain you do after the fact, or you have like an all DNA stain or whatever. But so you can do that. You can like stick it when you're making the gel. You can like stick the stain into your liquidy stuff, and actually cast it with the stain in it, and then the DNA can pick it up along the way. Or you can use like a buffer that has it in there like we do, or you can just like soak this gel in like a solution containing the stain. So a lot of different ways to do it. But yeah, so but remember that the dye that you see while it's running is just a tracking dye. So that's not going to tell you where your protein is. So be careful. Don't think that's it. But yeah, it's a good sign that it's moving. Yeah, and so another thing that's in this, in here is a tracking dye. So when the gel is running, you're going to see just this tracking dye. So we use this like Easy Vision, whatever. And so it has these tracking dyes so that you can see the progress of the run while it's going. This does not show you where the DNA is. It only shows you like an idea of where the dye front is. So, and then you can compare that to like what size it would correspond to in general. And also it's going to depend on the mesh size. So if you add more agarose, you get a tighter mesh. Less, you get a looser mesh. So a lower percentage of agarose is better for bigger things. A higher percentage is better for smaller things. So normally we do about a 1% gel for when we're analyzing PCR products of like a few thousand kilobases. So yeah, so the ladder here, you can see this is in like kilobase pairs. So on the right, you can see, sorry, I can't figure out how to make it so that you can actually see my pointer. But so on the right, you see like the visible dyes, and that's what you're gonna see when the gel is running. On the left, you have the, what happens when you have a fluorescent stain. So in this sample buffer that we use, it has a built-in fluorescent stain. But you can also fluorescently stain afterwards by soaking the gel in a fluorescent stain, or you can make your gel with the fluorescent stain. So what do I mean by fluorescent stain? So fluorescence is basically where you shine a light of one wavelength at something, and it shines back at a different wavelength. A couple common nucleic acid stains we use are ethidium bromide and DAPI. So DAPI, they just bind DNA in different ways. So ethidium bromide has gotten a really bad rap over the years, but because what it does is it actually like intercalates in between stacked bases of DNA, so it like wedges in between them. And this can cause problems if this was like in your body, because it can cause the like DNA copying machinery and stuff to make mistakes. But it turns out that ethidium bromide is really not, you'd have to have like basically drink a ton of it in order to have any negative effect, and so-called like safer stains aren't really much safer usually. So yeah, just follow good lab technique when using it. Wear gloves, wash your hands afterwards, the usual stuff, no matter what stain you use. Yeah, so something to keep an eye out for as well is that, so with protein, so with SDS-PAGE, one of the things that SDS does, in addition to add the negative charge, is it unfolds the proteins so that they stay linear, and you separate them based on their length instead of like their shapes, which would get in the way of them moving through the gel. So with DNA, usually, so if you have like linear DNA, it's not an issue. But if you have like a plasmid, so a circular piece of DNA, it's going to travel weirdly. So basically, if you have like, if the plasmid is super coiled, so this is kind of like if you have a phone cord, like one of those curly phone cords that is all coiled up, we call it supercoiling. And basically that makes it like a really compact little ball that can just like whiz through the gel. So that's going to travel faster. If you have like a plasmid that's nicked, so it's not super coiled, but it has like one strand. So basically when it's super coiled, it, like, because it has all this tension. So if you have like a nicked plasmid where the tension is relieved, it's going to travel slower than the linearized version, because it's kind of just like this weird sluggish like pancake thing. Not pancake, but it's kind of like an awkwardly shaped and hard to get through the gel, whereas the linearized can kind of like move snake-like through the gel. And yeah, so this is just showing you like what you would see in the case of nicked, linearized, or supercoiled DNA. And yeah, so that's basically all I wanted to say about Agarose gel electrophoresis, and there are more pictures and text and stuff on my blog. So good luck. Oh, and make sure that you run to red. You always want to make sure that you're putting the positive end of the gel where you're loading the wells, or sorry, where you at the bottom of the wells, and then the negative end where you're loading, so that the DNA gets repelled from the start to the end. And since it already has that negative charge, you're good to go without SDS. But you need that buffer to hold it down and give it color. So happy experimenting. See the blog version, and there's also a bunch of stuff on there about SDS-PAGE. So yeah, hope it helps.